This presentation gives you a flavor of the DNA Lab workshop. The DNA Lab workshop equips you with skills involved in making strategies in molecular biology. The workshop starts with calculation exercises. For example, you want to amplify mouse caspase 1 gene in PUC19 cloning vector using PCR. Typically, when you order the kit, you get a list of contents and a sample assay system. This assay system gives contents in millimolar, micromolar, enzyme units, and microgram quantities. You will learn to calculate and generate correct assay system for your need. Of course, typical PCRs may involve multiple genes and multiple tissues. You will also account for various controls. At the workshop, you will learn to design such complex experiments. We will read DNA sequences and learn to find probable genes in bacterial DNA fragments. It gets little more complicated with eukaryotes. Who doesn't want to decode secret messages? Here you would decode names of celebrities encoded as genes. You will play a game of encoding secret messages as gene sequences to challenge rival groups. Let's imagine that you joined a lab that works on Myxococcus xanthus and find that cloning vector PNG10A is extensively used for its molecular manipulations. We will learn to use softwares to draw plasmid maps and to extensively manipulate them for cloning experiments. Let's say you want to clone GFP gene of jellyfish into a cloning vector PUC19. We will learn to use PCR-based cloning strategies to solve such problems. Various academic and industrial research labs are interested in protein expression. You will simulate expression of proteins using PET vector series. In this exercise, you will learn to read the sequence of expression vector literally nucleotide by nucleotide and clone the gene of interest in the right reading frame. For example, here the gene with ATG as the start codon is cloned out of frame. This will result in expression of nonsensical protein. You will learn how to rectify such cloning procedures. At the DNA Lab workshop, you will learn the nuances of running and analyzing DNA gels, how to quantify the molecular weight and amount of DNA in bands during gel electrophoresis by image analysis.